Hi, I'm Clarissa. I'm been working in Griffith in Australia, and I'm going to talk about the molecular response of cnidarians to environmental stress, but I'm actually want to focus on the use of gene and protein expression as tools to analyze those responses. And this work has been done in collaboration with Dr. Kylie Pitt and Dr. Carl Mendugo. So as Nicholas has nicely presented right before me. It was really convenient. There's a lot of stuff that we know about how cnidarians respond to environmental stress, but there's still a lot to be done. And usually when we do study response of cnidarians to environmental stress, we use physiology, metabolism, and behavior variables. But this can be manifested after changes in the molecular and cellular level. So if we could use gene and protein expression as tools to analyze those responses, they often precede and mediate changes in physiology and behavior, and therefore they can detect more subtle and earlier responses to stress. But gene and protein expressions can vary under different conditions of stress. So for example, under different intensities and durations of stress. And they also analyzing and performing those the genetic things can be really costly, mostly transcriptomic analysis, if some of you are aware of it. So those things can also limit the amount of samples that you use in an experiment, so that you have to carefully design the experiment when you're thinking about using those tools. And when I say that, I'm just going to give a quick example of a hypothetical situation. And we have, for example, 3 different species. And let's say that the genetic profile of total gene expression for this species is something like this. So there's a sensitive species that under stress is gonna, they consider that they all have the same baseline of gene expression. So the sensitive species would not go back to the baseline after the environmental stress, while the tolerant and resilient species, for example, will have a very quick response to the stress and then quickly go back to the baseline. And the tolerant but not resilient species would slowly respond to stress and slowly go back to the baseline of gene expression. And if I have, for example, a limited amount of samples that I can use for my experimental design, and I sample here at the beginning and at the end of the stress experiment, I might consider the tolerant but not resilient species as a sensitive species to the stressor. And if I do a short experiment, for example, so if I reduce the time of exposure to the stressor and sample the beginning and end, I also might consider the tolerant and resilient species as a sensitive species to the stressor. So we have to be careful how we use this tool, because this all can generate a lot of inconsistency on data and regarding the rate of response to stress. For example, in this case, thinking about time. And that can lead to misinterpretation of resilience and plasticity of the animals to the stress. So the main objective of this part of my project was to examine how studies that use gene and protein expression to measure environmental stress response in cnidarians are being designed. So for that, I did perform a systematic literature review of cnidarians and cnidarian gene and protein expression studies related to environmental stressors, published into the end of the last year. I used the Web of Science to the search, and regarding the relevant words, I looked in the titles, author keywords, and abstract of the publications. It ended up with 781 publications that, after filtering, ended up to total of 307 publications. And for that, I analyzed the taxa that have been studied, the stressors that were studied, duration of exposure to the stressors, intensity of stress, period of acclimation and recovery, and the purpose of study, among other things that I don't have time to talk here today. And the general look of this data, it's basically, as you probably would expect, there's a lot of information on temperature stress. So about half of the studies were focused on temperature. And there is just 1% of those studies that were focusing on jellyfish, and that would include actually only scyphozoans, because the hydrozoans that you see here, they are only corals and hydra. And additionally, so just taking a little bit more into what those mainly coral actually studies were showing, and maybe we can learn something about them. The duration of experiments was significantly different when I compare between different purposes of research, but not significantly different when I compare between stressors. And either way, there's a majority of studies that do short experiments, but you can also see there's a very great variability on the experiments that we see here. The stress intensity was also not significantly different between different justifications that the authors give for why they use those intensities. And so this is something that I was a little bit impressed with. There's a huge variability also in intensity used by the studies. And duration of acclimation was extremely variable as well. And here again, we are talking a lot about thinking about corals, and usually they use visual, just the visual side of the coral as a health condition. So if they think the coral is healthy enough, they say that it was enough time for acclimation. And this is something that it's also problematic if we think about gene expression, as I mentioned before, because sometimes the profile of gene expression cannot be the same thing as you see visually, as the response can be very rapid and very different. And also, additionally, we have that 38% of the experiments include sampling during stress exposure. So not only at the beginning and end, so it's not that many. And only 12% of experiments sampled after, so during a recovery period, to analyze recovery. So taking all of those, some of the things that I'm pointing out here, we can see that there is an underrepresentation of cnidarians diversity. So it's a very little information on jellyfish in general. And also, there's an inconsistency of data generated regarding the rate of response to stress, and also the intensity that we saw that. So this can again lead to the misinterpretation of resilience and plasticity that I mentioned before. So if someone is intending to use gene and protein expression to analyze the responses, I suggest maybe trying to understand a little bit the plasticity and resilience of the animals that we're working with before performing some experimental designs. And for that, we can include, of course, if you have money, if you can do it, and tissue and animal available, you can include sampling during the exposure. You can sample during recovery and acclimation. But if not, you can perform pilot studies sometimes before performing your studies, just to elucidate a little bit more the rate of response to stress. And I want to thank all my colleagues in the lab that I'm here and the past labs that I've been through, and I could take any questions.